Welcome to the video abstract for our presentation at the 2022 meeting of the American Geophysical Union. This video is based on a conceptual analysis published at Frontiers in Water, info provided in the description box below. Our story begins with a common problem. Humans being fascinated by something in nature that's difficult to study directly. For a forest canopy, this system is literally out of reach. On top of that, its scale and complexity exceeds the physical limitations of our human sensory system. Indeed, various surfaces of the canopy at various times are exchanging water with the atmosphere, excreting some materials, hosting other plant or plant-like life forms, as well as many animal residents and visitors. On top of this, all these residents and visitors have some life cycle and processes of their own, and of course, the canopy surfaces themselves change as the trees go through their own life events. This circumstance probably sounds familiar to most in the natural sciences, and if you're a hydrologist, it probably sounds like the challenges inherent to studying watersheds. So several decades ago, hydrologists came up with a clever solution to studying the goings-on of watershed systems. They noticed that the precipitation that drains over the landscape can pick up signals from various watershed processes, then deliver them all to a single drainage point, the stream discharge. Thus, hydrologists have sampled stream discharge to search for all types of signals from the watershed, including isotopic signals, pharmaceutical compounds, naturally occurring organic materials, and so on. This brings us to the central thesis of our paper. Just like watersheds send signals via the streams that drain from them, plant canopies may send signals via the waters that drain down their surfaces. The precipitation and condensation that drain down tree canopies all the way to the base of their stems is called stemflow. And this is arguably as easily sampled by us surface dwellers as stream discharge. If stemflow carries signals, where would they come from? One way would be by washing signals off of canopy surfaces. In this way, stemflow can carry atmospherically deposited materials, yielding possible insights into plant-atmosphere interactions like air pollutant removal by canopies. It may wash canopy microbes down to the surface. That would yield possible insights into the dynamics of this so-called phylosphere community, which can be important for canopy health or nutrient cycling. Perhaps it washes off larger organisms or their excretions, sending signals about this community's composition or health too. Stemflow may also contain materials washed from the plant residents, like seeds, pollen, or in the case of lichens, soredia. Another way would be for stemflow to receive signals from within the plant. Some chemicals may be leached from leaves and bark into stemflow, yielding insights into canopy nutrient cycling. Perhaps the canopy supplies materials to draining stemflow a little more directly, like from these extrafloral nectaries, signaling important phenological events to scientists who have collected stemflow at the surface. When wounded, the canopy can release materials that may enter stemflow, like this image of a wound which is leaking alcohol flux. Pathogens may also be suspended within stemflow, suggesting an infection may be starting or be underway, even if visible signs have not yet appeared. Thus, some signals in stemflow may be distress signals from the canopy and potentially aid in tree conservation efforts. Hypothetically, if the scale insect and the fusarium fungal spore shown here were detected in stemflow at concerning concentrations, it may motivate conservation measures. Similarly, diseased or deceased animal residents may impart distress signals to stemflow. For example, could the cryptococcus pathogen that afflicts koalas be observed in the stemflow that makes it to the surface beneath them? Since the signals in stemflow originate primarily from the washing of one tree canopy, stemflow may be useful to assist in health monitoring for culturally valuable trees, like this old fella here. Now, there's certainly more in the publication, but in the end, our conceptual analysis left us wondering how much information a tree might passively share with us in their stemflow. The hypothetical possibilities listed here may just be the start. As stemflow itself currently has quite limited empirical relevance to hydrology, ecology, and biogeochemistry, perhaps future work on the possible signals it carries may motivate more natural scientists and even applied researchers to rigorously monitor this oft-ignored water flux. Thanks so much for watching, and please check out the open access special issue in which this analysis was published in Frontiers in Water.